Hello, welcome back to another video on cellular respiration. This video is going to be covering the linkage reaction and the Krebs cycle. The reason that I'm lumping these together is because these are the two where we're going to see CO2 released. And so if you watched my last video talking about anaerobic activities, you would have seen CO2 released during alcoholic fermentation. Remember that that is going to be anaerobic. Now we're talking about aerobic activities. So let's see what I'm talking about. So remember that in cellular respiration, it proceeds through 4, sometimes 3 steps. I sometimes call the preparation step the linkage reaction. I kind of consider that step 1.5. And so what you should notice here is that glycolysis is anaerobic. It happens in the cytoplasm. The other ones are aerobic. So that's what we're talking about for this video. We're going to be talking about steps 2 and 3 here. And I want you to understand that this is only going to happen if we have oxygen present. Now, oxygen is not directly involved here, but when we talk about the electron transport chain, we'll talk a little bit about why we still need oxygen for the Krebs cycle and the prep step to proceed. Okay, so looking at this, just remind yourself of last video, what we talked about in class. So we start with glucose. If we go through glycolysis, splitting the glucose in half, lysis of a glyco, meaning a sugar. Then we end with pyruvate, which are 2 3-carbon molecules. Counting my carbons, I have 6 carbons in glucose. I still have 6 when I have 2 3-carbon molecules. Now, pyruvate can go 2 directions. If there's no oxygen, then it will go and do fermentation. I can't live a long time without oxygen, so this is a short-term solution. And the prioritized pathway, if there's oxygen, is the aerobic pathway. So this is what we're talking about. We're going to be starting with the linkage reaction and the Krebs cycle. Okay, so if glycolysis is anaerobic and it happens in the cytoplasm and everything else is aerobic and is happening in the mitochondria, well, I've got to get my pyruvate into the mitochondria. And so the way that this happens is through the linkage reaction. So in this case, pyruvate needs to get into the mitochondria. And there's a really nicely named enzyme here called pyruvate translocase that is going to do that. That is going to get pyruvate into the mitochondrial matrix where the rest of the linkage reaction occurs. Now, I also need to convert pyruvate into a molecule called acetyl-CoA. Let's talk about that. The enzyme here is pyruvate dehydrogenase, and it's very important for regulation purposes, but let's talk about this process. So, the prep step, the linkage reaction, or pyruvate decarboxylation, is this idea of converting pyruvate into acetyl-CoA. Now, you should be able to count carbons here. Pyruvate, we know, is 3 carbons, and here they are, 1, 2, 3. Acetyl-CoA has 1, 2, only 2. And so that should raise alarm bells. I'm not able to create matter, so where did that other carbon go? Well, it's going to leave as CO2. So when any time that we see a change in carbon numbers, you should be thinking, where did that carbon go? And for our purposes, it leaves as CO2. Now the other part here, I'm breaking a bond. I'm breaking a bond, and we should remember that bonds store energy. And so that energy now is going to be stored in NADH. That'll be very important later on when we get to the electron transport chain in the next video. Okay, so I'm talking about this molecule called acetyl-CoA. Why are we converting it to acetyl-CoA? Well, that's because that's all that the Krebs cycle will really take. So we have to go from pyruvate to acetyl-CoA because it won't take pyruvate. We have to convert it into something that the Krebs cycle wants. And so let's look at acetyl-CoA. So the acetyl group is actually really small. It's only this portion right here, really small in comparison to the coenzyme A part, which you of course don't ever have to memorize the structure, but I do want you to understand that in comparison it's quite large compared to the acetyl group. And I want you to understand what a coenzyme is, which is just something that's going to help an enzyme function. It is not an enzyme itself. But it will help an enzyme function. We do see a little bit of this NCC pattern that's characteristic of amino acid chains, but then we have all of this that is certainly not protein-like. Okay, So I told you the whole point to getting to acetyl-CoA is so that it will be taken by the Kreb cycle. Now the Kreb cycle has lots of different names. Most biologists are trying to use Krebs cycle less, but it's still very popular in textbooks, and so most people still refer to it as the Krebs cycle. But you might also see it as the citric acid cycle or the TCA cycle for tricyclic acid cycle. This also occurs in the mitochondrial matrix and is going to do something that we've already seen, substrate level phosphorylation. We saw this in glycolysis, is happening here as well. Now I also want you to start thinking about numbers. Glucose is going to be split into 2 pyruvate, which each will turn into an acetyl-CoA. So we have 2 acetyl-CoAs as well. So that means that we are going to turn twice to be equivalent for 1 glucose. So as we are looking at this, I don't want you to think that you have to memorize everything. That would be very, very difficult at your level. Now, if you take biochemistry, you're going to eat my words and you're going to have to do a lot more. But I do want you to understand a couple of key parts here that will help you understand the process. So the first thing is we have the starting molecule that we've already talked about, acetyl-CoA. Acetyl-CoA is 2 carbons. It is going to be bonded together with oxaloacetate. Oxaloacetate and acetyl-CoA will come together to form citrate, which is 6 carbons. We've seen this type of cycle before, or something similar in the Calvin cycle. Now that was a 5-carbon RUBP and 1 carbon CO2, but the idea of 2 carbon molecules coming together so that we can regenerate later is important. Now let's count carbons. Citrate is 6 carbons. It's going to eventually be converted into ketoglutarate, which is only 5 carbons. So that tells you we have a change in carbon numbers. Anytime I see that, I should be thinking CO2 leaves, and we say bye, and we forget about it. Now remember, we broke a bond. Anytime we break a bond, we have energy, and that energy can be stored in NADH. And we'll talk about that later when we get to the electron transport chain. The similar thing is happening here. Ketoglutarate is 5 carbons. It is going to eventually be converted into succinate. Succinate is 4 carbons. That means that we have a change in carbon number. That means that CO2 leaves, it's lost. And that energy from breaking that bond can be stored as NADH. Now, there are some very cool complexes, very cool enzyme complexes that are important to the Krebs cycle if you go at a higher level of biology, but I do also need you to understand that ATP is made and FADH2 is made and an additional NADH molecule. So realistically, what you are responsible for this slide. It seems like very little in comparison to what I was just talking about, but it can be very difficult to start understanding these numbers if you don't see the process. So for 1 glucose molecule, this is for glucose, you will get 4 CO2, 2 ATP, 2 FADH2, and 6 NADH. But that's with 2 turns. So what's pictured here is acetyl-CoA. So for acetyl-CoA, you will get only 3 NADH, 1 FADH2, 2 CO2, and 1 ATP. This is important. So I could ask you potentially, what are the products for a glucose molecule, or what are the products for an acetyl-CoA? Now, some of these should make sense to you. Remember, acetyl-CoA was a 2-carbon molecule. Cellular respiration is the complete oxidation of glucose. We are going from a 6-carbon molecule down to 6 individual or single carbon molecules. And so we did most of that by going through glycolysis. We break it into 2 3-carbon molecules. Then we go from pyruvate to acetyl-CoA. Pyruvate is 3 carbons. Acetyl-CoA is 2 carbons, so 1 had to leave as CO2. That means now we have a 2-carbon molecule as acetyl-CoA. Both of those have to get oxidized to CO2. So this number should make sense because we had a 2-carbon molecule, so that means that we are going to convert into 2 1-carbon molecules. Okay, so let's review everything that we've talked about so far so that you are ready for the final step of cellular respiration, which is oxidative phosphorylation. So we start with glucose. Glucose we get from eating food. So we want to follow the complete oxidation of glucose. So glucose is broken down into 2 pyruvate molecules. Each of these are 3 carbons each, which means that we have not changed carbon numbers, so you should not see CO2 in this step. However, we do get 2 ATP out of it because we invested 2, but we get out 4. And so we get a net of 2 ATP through a process called substrate level phosphorylation. We also get 2 NADH, which will become very important when we get to the electron transport chain. Now, glycolysis happens in the cytoplasm, but everything else, assuming that there's oxygen available, is going to happen in the mitochondria. So this pyruvate needs to get into the mitochondria and is going to use an enzyme called pyruvate translocase. And once it gets in there, it's going to do the linkage reaction, which is going to convert pyruvate into acetyl-CoA. Now, acetyl-CoA is only 2 carbons. And so that means that we do have a change in carbon numbers, which means that CO2 is released during this step. Now, the enzyme that carries out this reaction is called pyruvate dehydrogenase. Now, acetyl-CoA is what the citric acid cycle wants, aka the Kreb cycle. So acetyl-CoA can enter the Kreb cycle, and through lots of fun chemistry, it is going to be completely oxidized, meaning that we are going to completely break down acetyl-CoA and release those carbons, those 2 carbons, as individual CO2 molecules. So the citric acid cycle releases 2 CO2 per acetyl-CoA. And during that, it also will produce 3 NADH and 1 FADH2 and 1 ATP. Now, that's per acetyl-CoA. Now it's going to happen twice because we have 2 acetyl-CoA. So we net 2 ATP, we net 6 NADH, we net 2 FADH2, and then 4 CO2. Now you probably are noticing that we do not get much ATP at all early in this process. And so really what we're stockpiling are these electron carriers. You will end up, before they go to oxidative phosphorylation, you will end up with 10 NADHs and 2 FADH2s, which are going to go to oxidative phosphorylation and do lots of really cool stuff. So I'll see you in that video. I hope your studying is going well, and have a good one.